Okay, good morning, everybody. And today we're going to finish off our discussion, our lengthy and complex discussion about the immune system. Now, I hope that by this stage of the week, you have all read the email that I sent and have all completed that worksheet and have set yourself goals that you need to set in order to get not just a decent, but a really good grade for your third class test, and then your final. Your study goals must be set and they must be adhered to. Otherwise, for many of you, there's going to be no hope of a decent grade. I have deliberately tried to go over this immune section slowly because I know how complex it is. But remember, please, that what you hear out of my mouth only gives you the skeleton. It is your first hearing. You cannot sit there like sponges and expect that it will all just magically soak into you. It won't. You cannot go into the exam or a test and squeeze the sponge and hope that enough dregs from the class hearing will dribble out so that you can answer the questions. It's not going to happen. The skeleton has to be filled out by you. It has to be turned from a sponge to a vertebrate that lives and functions on its own. Okay, so in any case, today we will complete our discussion about immunology, and I have already set an assignment. It is a relatively short assignment because it only covers immunology. So combine that with your studies for your last class test, and you'll do well, at least on this section. Do the same, use the same techniques for everything else you need to study. Your third class test is coming up. So you should be well on your way to already revising and studying for that final test, for that final class test. It's not your final test, it's your final class test. Okay, in addition, on next Wednesday, we will have a class quiz, but it will be a class quiz 10 questions, not the usual quizzy 5 questions, but 10 questions. And that will be on all of the material that we have covered since your last test. Okay, so with all of that under our belts, let's carry on our discussion. Probably one of the most important aspects of the adaptive immune system and how it works. And that is its ability to produce memory and how that memory benefits us. So here in this diagram, again, is encapsulated the entire story, really, of the activity of memory in the immune system. So let's have a look first at this line here. This is a response to a single antigen, and that is antigen A. So this is a measure of the amount of antibodies to antigen A that are present in the blood. That we can measure. It is done the whole time as a measure of the strength of immune response to particular pathogens, for example. So here, this is the antibody concentration here along the side, and notice that it's in log units. So have a look at how this curve is actually constructed. When we have our first exposure ever to an antigen, a pathogen with all its epitopes on the outside, et cetera, et cetera, et cetera, the system has to recognize that that pathogen is present and respond to it. So we saw that there's an innate response, which is very rapid, and it tides us over, and very often is enough to end an infection, get rid of a pathogen, get rid of an antigen. But if the adaptive immune system is brought into play. It actually takes time. The innate response is immediate. The adaptive response takes time. Have a look here. Here's our initial, just let me do one thing, please. Right. So here's our initial exposure here to the antigen. Maybe you just got infected with a virus of some sort or whatever. And how much adaptive immune system activity do we see? Practically nothing for up to 5 days. Now we've got to ask ourselves, what is happening in those 5 days? Why is there this silence, this lag? The answer is that you do not necessarily have the T cells and the B cells with receptors specific to epitopes presented by this antigen. You may not have them at all in your lymphatic system, but your bone marrow is continuously generating new lymphocytes, new B cells, new T cells. They are not very long-lived. Even in the lymph node, they're not very long-lived, so they are continuously replaced. And when they are replaced, they're replaced by B and T cells with randomly generated receptors. Each cell, each lymphocyte responding only to one specific epitope. It may take up to 5 days or so to begin to generate those cells at all. That's why we see no activity, because there are no cells to complete that complex cycle that we saw before of stimulating the T cell, or the dendritic cells stimulating the T cells interacting with the B cells, and then the B cells producing their antibody. Well, those cells just aren't there until some time has passed and they have been generated and they have lodged in the lymph nodes and begin to encounter one another. So the whole response can now begin. Here it is beginning here. At day 5, they're starting to interact with one another, and the production of antibody is just beginning. And it proceeds logarithmically pretty much over the next 5 days or so. So by the end of maybe 10 days, you are actually producing the working of an amount of antibody which is sufficient to begin really working properly. You will continue to build up the concentration of antibody till over 2 weeks or so. And then the antibody begins to overcome the infection. It begins to neutralize the viruses that you have inside. It begins to kill off those cells that are infected, et cetera, et cetera, et cetera. And by the way, I should tell you that this period here, these are times when you may actually feel really awful, really, really awful, because one of the things that happens when the adaptive immune response gets going is the body is stimulated to release all sorts of hormones which circulate, which cause things like high temperature, muscle ache, joint pain. All of these things are results of immune activity. So meanwhile, in the background, the antibody is doing its job and is wiping out the infection. The antibody binds up it all up. The macrophages eat the bound up stuff, et cetera, et cetera, et cetera, and the infection begins to die down. Eventually, the response, the immune response, is actively suppressed. You don't want this hanging around longer than necessary. It might begin to do damage to your own cells. So the antibody and the entire response is shut down when it is no longer needed. When the antigen is no longer circulating, this response stops. And here it more or less ends here. But in the meantime, those cells, immune cells, each of them, the T cells, T helper cells and cytotoxic T cells, and the B cells, in addition to forming their effector cells, the cells that do the work, they also generate memory cells. And those memory cells have on their surface receptors for the same epitope, but they are silent. They do not act in this period of the adaptive response. This is called the primary response. And it specifically, of course, produces antibodies to an antigen. But it may, and I should tell you, that it doesn't necessarily just produce one antibody. It may produce a whole suite of antibodies to different epitopes on the same antigen. The end result is the infection is vanquished. The immune response is tamped down and stopped, but the memory cells remain. And they may remain for a really long time, for like 30 years or more. And they may just remain silent, inactive for all of that time until the person dies, if they never called on again. But what happens if we expose this person to the same antigen or to the same epitopes? Right? Immediately, the memory cells react. And they do so in two important ways. This is called the secondary response. And remember the different groups of memory cells we have. First of all, let's think about the memory B cells. The memory B cells, when they encounter the antigen a second time, immediately begin to activate. The memory T and helper cells, they, when they see the antigen presented again by dendritic cells, they immediately begin to clone themselves frantically, and their activity is heightened, so that the amount of time that it takes them to meet up with B cells, which are already beginning to clone themselves, is very short. So then, in addition, we have memory cytotoxic T cells, the same story. When they see the antigen presented by dendritic cells, or when they see it presented by cells of the body, they immediately react. There are these two shortcuts, B cells immediately being activated, and then the memory cells being activated, so that the entire community of the adaptive immune system immediately begins to react. It doesn't have to undergo this lengthy period of producing the cells and then getting them to the lymph node and having them meet. That is all short-circuited. And we have an almost immediate response. Here is a second exposure to the same antigen. And you'll see that within days, often within hours, in actual fact, there is a secondary immune response. And look at the strength of it. Not only is it rapid, but it is extremely strong. And the B cells produce a prodigious amount of antibody. There's something else. I didn't discuss it I referred to it, I think, but I didn't discuss it in any detail. As the adaptive response proceeds, the B cells actually produce new and different versions of the same antibody. The antibody is improved on continuously. And antibodies produced which works better and better and better and better. And also different kinds of antibody with different functions are produced, all to the same epitope. When this dies off here, the memory cells that are left here are the memory cells that carry the very best antibody, potential for antibody. They are memory cells of the most highly improved B cells. Now, when the secondary response starts, they carry on this process of refining the antibody, making it more and more. When eventually the secondary response tails off, which it will do, but often only after a long time, the memory cells that are left from this are even stronger. They're even better. So just for a start, let me point out that this is one of the reasons why booster shots for vaccines are often very important. Because each time you have a booster, you improve the adaptive response in this way. Okay, so that's our response to antigen A. We get a primary response, and then we get a secondary, very strong secondary response because of the memory cells, these highly efficient memory cells that remain from the primary response. So let's ask the question, at this point here where we re-expose to antigen A, what if we include another antigen? What if we put antigen B in here, a totally different antigen, and see what the immune system does? Well, if we monitor the antibodies, we see exactly the same pattern as we saw here. Having this very powerful response to this particular antigen, A, makes no difference to the response to antigen B. Antigen B still has to be processed, as any antigen that has never been seen before must be processed. So here, in fact, is a primary response to antigen B. And then at the end of when all of that has been voided, memory cells remain to antigen B as well. We carry memory cells for thousands of antigens, thousands. And we're capable of generating millions of responses to incoming antigens, to new antigens that our adaptive immune system sees. That's why it's the adaptive immune system. It adapts to new incoming antigen, and it adapts with enormous specificity. Okay, is that all I need to tell you? I think so. Okay, so just a couple of other things to talk about antibody. Antibody actually can be secreted into various body fluids, and especially it can be secreted into mucus, into the mucus barriers of the body, where it can stop the activity of pathogens, etc., before they even get into the mucus linings of the organs. It can also be secreted in breast milk, and it can also pass across the uterus, across the placenta, sorry, in the uterus, into the fetus. So there are various ways now that this antibody can begin to provide protection outside of our own blood circulatory system. In the case of a breastfeeding mother, she is continuously passing antibody, which she is producing, to her infant. And this provides the infant with a certain amount of protection during the most vulnerable time of its life, which is the first 6 months or so, when its own immune system is not yet mature. So this is known as passive immunity. And it provides immediate protection, but it is relatively short-lived, but very important in infants because it provides them with that protection. It's one of the reasons why breastfeeding is so important and so beneficial. But passive immunity, there's another way in which it can operate, and that is we can purify antibody from people who are recovering from an illness. This has been done in the COVID epidemic. The people who are recovering from COVID act as antibody donors. And the antibody is taken out and purified and then injected into another person who is still suffering from the disease. And it's not always that successful, but it will provide, in many cases, it will provide at least a certain level of protection, maybe protect them from very serious illness or help them recover quickly from serious illness. That is another example of passive immunity. Okay, so I say active immunity obviously is something which develops naturally. Immunity refers to our ability to use the adaptive immune system to fight an infection, to fight an antigen. So the most common way in which active immunity is maintained is through memory cells, and they are degenerated, as we've said, to an antigen to which we've been exposed in the past. But the development of that immunity can also be induced artificially, and this is the basis of vaccination. Now note that vaccination is completely different to donation of antibody. Donate antibody, you're just donating a substance in solution which acts to knock down the antigen. In vaccination, we're actually training the immune system to respond. What happens in vaccination is that the body undergoes this. But usually it is actually quite a silent response. We don't have all the illness and everything that's associated with the real pathogen. Instead, there is a mild stimulation of the adaptive immunity to a specific antigen, maybe a dead virus or part of a virus or something like that, where we train the immune system to recognize that as an antigen that it has seen. Then if in the future, obviously it makes memory. In the future, if it ever sees that antigen again in the form of a new infection, the immune system responds immediately with the secondary response and wipes out the pathogen, deals with the antigen before we've usually even know anything about it. Now that is the principle of vaccination. Vaccination is a form of active immunity. Just to say that I've already just alluded to it, that there are many different ways in which somebody can be vaccinated. The earliest vaccination of all was which was done by inoculating people with weakened or smallpox virus. I told you that story. The next development in the vaccination story was the discovery that people could be vaccinated with an organism or an antigen which looked very much like a pathogenic antigen, but which was harmless. So in the early days, smallpox vaccination was done by injecting them with cowpox virus, which is essentially harmless to human beings. It's actually pretty harmless to cows, to tell you the truth. But it's harmless to human beings, but it looks sufficiently like smallpox that the immune system confuses the two. And a recovery from that primary infection of smallpox, of cowpox virus, gives lifelong protection from infection by the smallpox virus. We can also vaccinate people rather, with organisms, pathogens, which have been substantially weakened. That is done in the laboratory. They are usually passed through serial infections, often in rabbits or sheep or something like that. And each time they pass through that infection cycle, the virus is weakened. we say it is attenuated. And the final vaccine product is a highly attenuated virus. It's a live virus, but it doesn't cause disease. Then the last way that I've just mentioned is we often vaccinate with dead virus or with portions of dead virus, maybe fragments of the outer coat or things like this, which provide enough epitopes that the primary response can be elicited. In the case of the COVID epidemic, the Pfizer and Moderna vaccines are both a new kind of vaccine, and that is an mRNA vaccine. And what the mRNA vaccine does is it introduces a piece of messenger RNA into our cells. For a very brief period, that messenger RNA is treated like any messenger RNA in the cytoplasm of our cells. It is translated. And the protein that is translated is actually a viral protein. It's one of the spike proteins that is on the outside of the virus. But it's produced in a form that allows it to be secreted into the bloodstream. And as soon as it appears in the bloodstream, it gets swept up through the body to the lymph nodes. It proceeds through the entire primary adaptive immune response. It's just that there's that added step at the beginning. We get our own cells to produce the protein. Now, for reasons that I don't actually know, it appears to be a very powerful vaccine. I'm not sure what the advantages are. I'm sure somebody's either worked them out or working them out. But it's important to note that that little piece of messenger RNA is very short-lived, probably the order of 20 minutes or something. But it produces enough protein into circulation in our blood that our immune system can respond to it. Okay, so let's talk now, just to finish off, about some of the things that can go wrong. Now, there are many things that can go wrong with the immune system. There are a whole host of diseases that arise because of errors in the immune system, especially where the cells of the immune system begin to attack our own cells, where they lose the ability to distinguish between foreign and self. But we're not going to talk about that. Those are the autoimmune diseases, and there are many of them. If you're interested, you can go and look up that term, autoimmune disease. But I want to tell you about a couple of things which are probably more familiar to you and explain them to you. The first is many people suffer from allergies. And allergy is termed a hypersensitive response. And it is a response of the immune system to antigens which are normally completely harmless, except that for some reason they elicit an immune response in certain people. And then they are termed allergens, things that will cause an allergic reaction. The allergens are almost always entirely harmless. Just think about it, grass pollen. What possible harmful aspect can there be to grass pollen? It can't grow in you, right? It can't do any damage to you. Why do we respond to it at all? And the reason is that that allergen is actually tricking a part of the immune system. It's fooling a part of the immune system. So we got to go right the way back to the very beginning. And I told you that one of the first cells, in fact, the first cell, which will respond to something like bacteria being introduced through a wound, or a wound being created, or damage being caused. The very first response, the cell which produces our initial responses, is called a mast cell. Mast cells occur throughout the body. They are present in all of our tissues. And one of their responsibilities is to release a set of chemicals, of hormones, which induce inflammation. And the most familiar one to us all is probably histamine. And histamine is what causes all of those symptoms of inflammation, like dilation of the blood vessels, causing leaky blood vessels, swelling, causing fluid buildup around cells, etc., etc. That's all the responsibility of the mast cell. And I won't discuss all the mechanisms, but I will tell you that this is a normal, healthy response. And it is a response, if it wasn't there, our entire immune response would be very, very poor. Mast cells are extremely important, although we haven't discussed their role in all that much detail. If you look at them, they're full of granules, and those are granules of histamine and other substances, which cause the inflammatory response. They also call in other cells to this area of inflammation to come and start doing their work. Very much a part of the innate immune system. However, mast cells have a very peculiar property, and that is that they can acquire, look at these, what is this? They can acquire antibody. They acquire antibody actually of a specific sort. It is called IgE. And IgE is produced especially in response to parasites. And this is one of the important roles of mast cells. They interact with another group of cells in the presence of parasites to cause inflammation and to also cause complement to be activated in an attempt to expel parasites from the tissues. So here's what they do. They can coat themselves with IgE. They coat themselves with IgE of all sorts of specificities. It's just if there's IgE around, it can stick onto the mast cell. But these variable regions here of the antibody, they can be, each of these antibody molecules can be to a different epitope. So this mast cell is forested with these antibodies, and they now act as a receptor. They act as a mast cell receptor. If, in fact, this epitope binds here, it causes the mast cell to just pour out histamine. And so that's its normal function. It's actually there mainly to pick up information about incoming bacteria, those sorts of things, but also to interact with parasite antigens. That's a perfectly normal part of our immune system, and it normally works absolutely fine. There's a problem, a big problem, though, with these receptors. And that is that sometimes it's largely genetically determined. We don't understand all of the details of it in actual fact. But for some reason, some of these receptors here can bind completely harmless substances. An allergen is actually something which will bind to a mast cell receptor. And let's say it's grass pollen. Well, why on earth would your immune system be mounting a response to grass pollen? It's harmless. It's just an accident. It's just that some of the epitopes on the grass pollen suit the epitopes on the mast cell. There are usually two stages to these systems. There has to be an initial response. That is when the mast cell becomes sensitized to a particular allergen. In other words, the allergen comes in and it causes a whole immune response. Normally it wouldn't cause illness, but the immune system produces IgE to it, and that forests the mast cells with IgE receptors for that particular allergen. That initial exposure, there's usually no bodily response. There's no real inflammatory response. It can be very, very brief. You don't need to produce much of the IgE in order to sensitize the mast cells to that particular epitope. If you never see that epitope again, you get no response. But remember what I told you. The mast cells occupy all of our tissues. So let's think about the grass pollen for a minute. If you're sensitive to grass pollen, what is your response when you breathe in or grass pollen is in the air? Your eyes swell, they tear up. Your nose begins to stream. All of these responses, what are those responses? They're inflammatory responses. They are inflammatory responses. And that inflammation is caused because this mast cell that has been sensitized to grass pollen now is seeing it again. And you've now got lots of mast cells that are sensitized, that bear this particular IgE for that particular epitope. And though all of those mast cells in the eyes and in the nose, where they're exposed to the pollen, release histamines and other inflammatory compounds, which cause the blood vessels to dilate, red eyes, blocked nose, etc. They cause fluid to begin to be leaked into the tissues. That's again the swelling, even the itching. That is a response. That is a response to compounds released by the mast cells as well. It's the mast cells mistakenly causing the inflammatory response and inappropriately. So that's fine. Look, hay fever, these sort of things. That's normally a nuisance. It's not a life-threatening situation. But I want you to picture something else. Your mast cells are present all through your body. And all of those mast cells could be bearing this IgE. The thing is, exposure to the antigen is localized. Your eyes, your nose is not circulating in your blood. But now just imagine that you've had an exposure at some point to something like penicillin. Penicillin is injected. All the mast cells of your body can be sensitized to the penicillin in an initial exposure. Now, at some point in the future, if you are allergic to penicillin, an injection of penicillin sends the penicillin into circulation through your body. And all of the tissues, all of the mast cells in all of the tissues of your body release their inflammatory substances all at the same time. Now what happens? All of the blood vessels, even your major blood vessels, dilate. That's what histamine does on a local scale inside your eyes. Your eyes go red because histamine causes the blood vessels to dilate. Put into circulation and put an allergen into circulation in the body, all of the blood vessels dilate. All of the blood vessels become leaky. That's another function of histamine. They become leaky to serum. So that serum pours out into the tissues. If somebody is allergic to something like penicillin and they get a penicillin injection after they have been sensitized by a previous exposure, they can be placed in a very, very dangerous situation. And this is called anaphylaxis. And you can picture what is happening. Just a few simple things. If all of the blood vessels of the body dilate, what's going to happen to your blood pressure? Your blood pressure is going to plummet. And especially, you're going to have difficulty moving sufficient blood to your brain. Your brain begins to get starved of oxygen. Second, the blood vessels become leaky. So the volume of blood decreases as the fluid passes out into the tissues, especially in your respiratory tract. The swelling of the tissues can be extremely dangerous because it begins to block airways, it begins to block your alveoli. Your alveoli begin to fill with fluid. Anaphylaxis is a life-threatening critical situation. There are other things which go into circulation in your blood, which can do the same thing. Bee venom, for example. If you are sensitive to bee venom, then the same situation can arise for the same reason. It causes all of the mast cells of your body to degranulate if you're sensitive. So these are ways in which a perfectly normal response is in a way perverted just by chance and by genetics to present a real danger. So that's the one that may be familiar to you. Oh yeah, of course, what is the first thing that you do? Oh, sorry, let me go back. If you have an allergic reaction starting, well, antihistamines. Why do you take antihistamines? Because that's what's causing all the problems. It's one of the major causes. It's not the only one, but it's one of the major causes of all of these problems. So you take antihistamine. If you go into anaphylactic shock, they will give you adrenaline, epinephrine. Epinephrine causes all the blood vessels to constrict, raises your blood pressure. They will immediately put you onto a drip to try and bulk out the blood volume, to raise your blood pressure. Then you're filled with antihistamines and all sorts of drugs to stop the leaky vessels, etc. Hold on, I've done something really stupid. Knocked out my power cord. No, all my gesticulations are knocked out the power cord. Okay, so there's the one that's familiar to you. Here's the next one that's familiar to you. This is also a hypersensitivity reaction. This is a reaction to poison ivy. Believe it or not, this is a very complex immune response. People tend to think that poison ivy has got something in it which just directly causes the swelling and the itching and the blister on the skin. That's not true. In fact, those symptoms are caused by your immune response. And this is essentially what happens. There's a substance in poison ivy. It's actually an oil, very similar to the cell membrane, the phospholipids and everything. It can pass through them very, very easily. So it passes into your cells, your skin cells. So you'll have your first exposure. You're working the first time you've ever seen poison ivy. You rub it up against your skin. You actually are unlikely to see any reaction at all at the first exposure. But two things have happened. The first is that the cells have absorbed the toxin. It's actually become intracellular. And so your cells will actually start to present it by MHC class 1. Now, initially, that does nothing at all because the adaptive immune system has not been activated. For the adaptive immune system to be activated, the dendritic cells have to also pick up, which they do very easily. It can just make its way into their cytoplasm through their membrane. They then process it as if it's part of the normal adaptive response, and they present it through MHC class 1. And they will set off a minor adaptive response to this allergen. You usually don't even notice it at all. But in the course of doing so, they form memory cells. And especially the most important of all are memory T cells. The most functional one being the cytotoxic T cell. Now remember what cytotoxic T cells do. They respond to MHC class 1. They respond to allergens, to epitopes rather, presented by cells. All of our cells can produce MHC class 1, and they can produce it with epitopes, which are normally derived from normal cellular processes. It's one of the ways our cells label themselves itself. But if a cytotoxic T cell sees something foreign, it will kill that cell. But it doesn't only kill the cell. it interacts with other cells of the immune system, and it activates a very powerful inflammatory response. So here's what happens. You get exposed, you have this little minor adaptive response, you form memory cells, which are very, very long-lived. The next time you're exposed to poison ivy, the memory cells are immediately activated. And in particular, the cytotoxic memory cells, cytotoxic T cells, which are memory cells, can respond directly to those cells of the skin which are exposing the poison ivy toxin. And what do? They kill those cells. In doing so, they spark a very, very powerful inflammatory response. The cells die, and masses of fluid are poured in around them through the inflammatory process, and you end up with these big, spectacular blisters, which are also incredibly itchy, and that is the responsibility of histamine and various other substances, immune substances as well. So that's the primary response. You usually don't notice anything. This is the secondary response, which can be really, really spectacular and very, very debilitating. So that is basically it. Now, this has not been a particularly long class, so this might be an opportunity for you, please, to go to your assignments and do the assignment while all this material is fresh in your head. And remember, please, next Wednesday, there is a quiz, okay? And so with that, I'll say goodbye for the day.